Hello. It's an honor and a pleasure to speak to the Hope for Rare Foundation about my own efforts to start companies, develop companies that are developing therapies for rare diseases. I'll talk about the founding of Genzyme and the development of an enzyme replacement for Gaucher disease. I'll talk about Tevard, a company that I helped found more recently to develop gene therapies for haploinsufficient genetic disorders, a type of genetic disorder that has not been widely studied, specifically to treat the childhood epilepsy developmental defects in Dravet syndrome. I've helped start many companies over the years. Many of them focus on treatments for rare diseases. And as I mentioned, I'll focus specifically on Genzyme and Tevard. But Carmine, Cargene, Cerberus, other companies that I'm currently working with are also developing very different kinds of treatments for rare diseases. I've been privileged to serve with Kevin Wang on the advisory board of the Chinese Organization for Rare Disorders. And I've seen firsthand the enormous impact that Kevin and his colleagues have had in making China a country that is aware of its own rare diseases and beginning to develop treatments and diagnostics for rare diseases that are common in China. Also, I've been on the board of trustees of Boston Children's Hospital for about 15 years, where I see firsthand the research undergoing there in developing first genetic understanding of rare diseases, and then secondly, treatments for many rare diseases, particularly those that affect Children's Hospital. And in parallel, I've served as chair of the advisory board of the Massachusetts Life Sciences Center, an agency supported by the Commonwealth of Massachusetts, charged with oversight of $100 million investments over each year for 10 years in developing life sciences in Massachusetts. So rare diseases affect as many as 25 million people in the United States and many more worldwide. There are more than 6,000 rare diseases, defined here as any disease or condition that affects fewer than 200,000 US citizens. Europe similarly has approximately 30 million people with rare diseases, and China somewhere between 50 and 82 million people living with a rare disease. As worldwide, a large fraction of the rare disease patients in China are under the age of 18. What's important to realize is that autosomal recessive genetic diseases, the most common kind, are concentrated in specific ethnic groups. Because of intermarriage, these genes can spread within a population such that a sizable number of individuals can inherit both defective genes, one from each parent. Just two examples here: unusual genetic diseases that are common in countries such as Iceland and Finland. And the United States has passed an orphan drug law. I urge governments worldwide to develop similar laws that encourage the development of therapeutics for rare diseases. Most importantly, in the United States, the FDA gives 7-year market exclusivity for orphan drugs. That is, if they approve a drug for a particular rare disease, they will not approve a similar drug for 7 years. this is not changing patent law, but rather market exclusivity. And there are other features as well. Even if one cannot develop therapies for rare diseases, it's very important to uncover the underlying genetic basis. Because many of these diseases are concentrated in specific ethnic groups, prenatal genetic testing of members of these groups can eliminate these autosomal recessive genetic disorders in 1 or 2 generations. And one group that has been extraordinarily successful is the Orthodox Jewish communities, particularly based in New York City, where every young person going through adolescence donates a DNA sample, and they test for a number of genetic diseases that more specifically affect this ethnic group, diseases like Tay-Sachs disease or Gaucher that I'll speak about in a moment. And these genetic diseases have been virtually eliminated in these ethnic groups within a generation or 2. So with this general introduction, let me speak first to Genzyme and then to haploinsufficient genetic disorders. Genzyme's first therapeutic is an enzyme replacement, a protein therapeutic to treat type 1 Gaucher disease. Gaucher is considered a lysosome storage disease. That is, the defect is loss of function of a particular enzyme that's localized to lysosomes that, like other enzymes in lysosomes, breaks down outworn molecules, defective molecules of all sorts into their molecular constituents. Here, it's missing an enzyme called beta-glucocerebrosidase that's essential for degradation of a particular glycolipid. And as indicated, it's most abundant in descendants of Eastern European, so-called Ashkenazic Jews. It's generally not fatal, but it's progressive, debilitating, and sometimes life-threatening. It affects mainly the cells in the body called macrophages, large cells that, as their name indicates, digest and degrade outworn body cells, invading pathogens and the like. And symptoms can appear at any age, but can include easy bleeding and bruising, anemia, weak bones because the bone marrow fills up with macrophages that have this undegraded fatty substance in them, bone and joint pain, and so forth. And 8 of us who were faculty at MIT started Genzyme around 1980. But credit has to be given to Henry Termeer, the founding president of Genzyme Corporation. He, not a scientist, but a business person, had the vision that one could build a company around developing therapeutics for rare diseases, building a profitable company, and in parallel, working closely with patient groups to make sure that the therapy was available to anyone that needed it. So Genzyme's first product was this enzyme replacement called Cerezyme for type 1 Gaucher disease. It's produced as a recombinant protein secreted from engineered Chinese hamster ovary cells. If one simply injected that protein into a person, it would circulate in the blood, but it would not get into the macrophages, the cells that really are missing the protein. So what we had to do was modify the carbohydrate chains that are attached to the recombinant protein to expose mannose residues that would bind then to receptors on the surface of macrophages. That is, when transfused into a patient, Cerezyme circulates in the blood, but then binds through this mannose receptor, mannose interaction to the surface of macrophages. Macrophages then internalize the bound enzyme, a process called receptor-mediated endocytosis. It's transferred to the lysosomes, where serozyme takes up residence and degrades the accumulated glycolipid. So again, serozyme, like most recombinant proteins, is produced in these enormous 1,000-liter bioreactors that grow these recombinant mammalian cells. And then purification requires the skills of many chemical engineers, but it's now become a routine process. So in summary, serozyme, really the first biotherapeutic for a rare disease, is a personalized medicine. It's a recombinant protein. It's a recombinant protein targeted to a specific cell type, and it's based on sugar engineering. So this is one of the reasons that I emphasize the founding of Genzyme in the context of developing therapeutics for rare diseases. There's another very personal reason. As mentioned, Gaucher disease is fairly common in descendants of Eastern European Jews. When our daughter was pregnant in 2002, well after I left Genzyme, she and her husband, being of Eastern European Jewish ethnicity, were offered a series of prenatal genetic tests, ones that would detect genetic diseases common in this population. And these tests showed, rather surprisingly to us, that both our daughter and her husband were carriers of a mutant Gaucher glucocerebrosidase gene. That is, the child she was carrying had a 1 in 4 chance of having Gaucher disease. Indeed, amniocentesis confirmed that the child she was carrying did have Gaucher disease. But because of serozyme, which was developed 15 years previously, we and our genetic counselors could determine that the child would be treatable with the drug his grandfather helped develop. Indeed, I'm pleased to report that Andrew, who's now 19 years old, has been on the Genzyme serozyme since he was 10 years old. He is doing fine. He recently completed, you can see him on the left, a bicycle trip across the United States. Importantly, he has never been sick a day in his life from Gaucher disease. Every other Saturday, he goes to his university's healthcare facility, brings a little homework, and gets infused with serozyme. And that will continue certainly for many years, but otherwise he lives a completely normal life. He's an athlete, and hopefully he will one day be a medical doctor. Genzyme, of course, based on serozyme, makes a number of other recombinant proteins that treat other lysosome storage diseases, and has been, of course, a very successful company. It was purchased about 12 years ago by Sanofi for about $20 billion US. So let me turn to a very different class of genetic disorders, which are haploinsufficient genetic disorders. And a company that I founded with 2 business people, not scientists, who have daughters with Dravet syndrome. Tevard, the name comes from Dravet spelled backwards. That is, we want to reverse Dravet syndrome. So these haploinsufficient genetic disorders are caused by loss of function, not of both copies of a gene, but of one, hence the name haploinsufficient. In the case of Dravet syndrome, the gene is SCN1A, which codes for a voltage-gated sodium channel that is widely expressed in the brain, but particularly abundant in inhibitory neurons. That is, in Dravet syndrome, there are not enough of these sodium channels in certain nerve cells to have the cell function properly. And as a consequence, there are a number of quite devastating consequences, including epilepsy that is very, very difficult to control, plus a number of developmental delays. Many of the children are not verbal, and they have muscle issues and many other things. Thinking of a gene therapy for a disease like Dravet syndrome is very complicated because it's highly regulated. It's expressed at very different levels in different cells. As I said, it's an ion channel that's involved in nerve conduction. And experiments in mice show that too much of the protein also causes disease. So the objective is to increase the level of SCN1 gene expression and of the sodium channel about twofold, such that it would be normal levels, normal in whatever cell the gene is normally expressed. There are drugs for Dravet that treat the epilepsy, but do not affect the underlying causes of the disease. So we founded Tevard to develop treatments specifically for Dravet, but it immediately became apparent that there were 700 other haploinsufficient genetic disorders. I've listed some of them here, and in the next slide, I don't expect you all to read them, except they are very common. None of them have treatments, certainly no genetic treatments, gene therapy treatments, and so forth. Just to put it in a framework, I pick one of the diseases because I worked with it before, Kleefstra syndrome. Very different type of effect. It's loss of one copy of a protein that encodes an enzyme, a histone methyltransferase, that transfers a methyl group to a particular position on a histone gene. This sounds very arcane, but this protein, by methylating this particular histone, tags it for gene repression, turning off RNA synthesis. And loss of half of these enzymes causes inappropriate gene expression and many developmental problems, speech impairments, mental retardation, and so forth. So I won't go into elaborate details, but simply point out that the technologies that Tevard has developed, gene control in the nucleus, that is, they do not affect messenger RNA synthesis or splicing or transport into the cytoplasm. What they do is enhance the translation of the messenger RNA to the point where the wild type copy of the gene can increase its output twofold. That's one iteration. And in the other, replace a stop codon in a messenger RNA and put in the normal amino acid. Let me just illustrate what I mean. So we have a whole program in what are called suppressor tRNAs. That is, between a quarter and a third of all genetic mutations in all human genes convert a codon for an amino acid, a so-called sense codon, to one of three stop codons, UGA, UAG, or UAA. These stop codons are, of course, normally at the 3' end of every protein coding gene and tell the ribosome that's making the protein to terminate and make a full-length protein. But when these codons, these stop codons, are in the middle of a gene, they make a truncated protein that is immediately degraded by the cell and, of course, is non-functional. tRNAs that read nonsense codons are not known in higher organisms, certainly not in humans. But using genetic technology, we have taken normal transfer RNAs, say a normal arginine transfer RNA, and changed its anticodon so it reads a nonsense codon, say UGA, TGA, and inserts the normal amino acid, in which case you make a full-length protein. So this does work for Dravet syndrome, certainly in cell cultures and in animals, and we hope to be in human clinical testing very soon. Here's an experiment in cell culture where if we express the mutant SCN1A, this one has an arginine to UGA mutation, which is actually found in one of the two daughters of my colleagues. There's no protein made at all. One can see it in this lane, but expressing also a gene for the arginine UGA suppressor, one makes normal amounts of the protein. And this protein, as expected, is normal function. It should have and does have the normal amino acid sequence. And in this whole cell patch clamp experiment, which I won't detail, it shows that the sodium channel opens and closes absolutely normally. So what we have done is put the gene for these this and other suppressor tRNAs in a adeno-associated virus, and we've administered it to mice, and we're beginning to contemplate experiments in non-human primates. This is another example. This is the MECP2 protein that is mutant in Rett syndrome, another haploinsufficient genetic disorder with somewhat different genetics because the protein is X-linked. But again, we are able to rescue the mutant SCN1. I'm sorry, we are able to rescue the mutant MECP2 protein, one that has an arginine UGA mutation, by expressing adeno-associated virus encoding our suppressor tRNA. So that's one set of technologies that we are developing that will treat a whole range of genetic disorders caused by nonsense mutations, particularly in haploinsufficient genetic disorders, where it will restore the normal level of the protein, normal in whatever cell it's expressed in. We also have other technologies that we're developing that enhance the stability of the wild-type copy of the message. That is, in these haploinsufficient genetic disorders, there is one functional copy of the gene, but its protein output is insufficient. And if we can double the output of the normal copy, we'll restore normal function. We have several technologies that do it. I'll just describe one for you. It's a protein RNA-based gene therapy. As you can see in this diagram, the protein consists of two components. One, a subunit that, guided by a guide RNA, targets the protein to a particular RNA in the cell. And the second component, here called PABPC1, if attached to a messenger RNA, will stabilize the messenger RNA. And it works. For instance, here we are again looking at Rett syndrome. We use it because we have a very clean assay for this protein. As you can see in the diagram on the left, co-expressing in cells that express a mutant MECP2 protein. I'm sorry, in cells expressing just the MECP2 protein. It could be either wild type or mutant. co-expressing the guide RNA and the relevant Cas-PAB-PC1 protein, we can increase the output of MECP2. And as the diagram on the right shows, by positioning this protein complex guided by the guide RNA at different positions in the 3' untranslated region of a messenger RNA, we can upregulate the stability and the protein output by several degrees, up to 3 or 4-fold. What we want, of course, is a 2-fold increase, and we're able to do it. Yet a third example is SYNGAP1, another haploinsufficient genetic disorder causing epilepsy, autism, and sensory processing defects. And here, again, in appropriate culture cells, we can see an upregulation of SYNGAP protein 2-fold. So in summary, Tevard, which is a young company, we are in Lab Central, one of the premier incubators in the Boston area. We are developing these treatments, and hopefully we will be in non-human primate trials shortly, and then afterwards in human clinical trials. But our goal is to develop these types of gene therapies for Dravet syndrome, for other haploinsufficient genetic disorders, and more generally for any type of genetic mutation that converts a sense codon to a nonsense codon. So I will end with photographs of 2 families. On the left is part of my family. You can see on the right of the figure, the tall young man, that's Andrew. We're all going on a hiking expedition in the desert, and despite his Gaucher disease, he's living a normal life. The young girl in the purple dress, in contrast, is the one with Dravet disease. She is not doing well, and hopefully at some point, gene therapy or perhaps other technologies that others are developing will cure her disease. So I will stop sharing my screen. I wish I could be there in person to answer your questions, but I'm quite happy to carry out correspondence with any of you who would like more information on the companies and the concepts that I talked about. Thank you.